يلا بسم الله الرحمن الرحيم. I think I will start right away. First of all, good evening to everybody, and I'm so happy that I can see around 52 attendees, which is good. So people are hungry to know about real-time PCR. So what I would like to introduce myself. First of all, my name is الزدجالي. I'm actually an associate professor in College of Medicine and Health Sciences at Sultan Qaboos University. There was an interest that a lot of fresh graduates and also a lot of scientists and also a lot of technologists, they would like to know about real-time PCR. So I was thinking like, why not to give like a small like a webinar, just trying to describe what is a real-time PCR. So this webinar is going to be an introductory webinar. And basically in this introductory webinar, I'm going to discuss 3 points. The first point is basically to understand what is a real-time PCR, because if you understand what is real-time PCR, it makes it easier to understand how it is used and what you can use it for. So we're going to discuss about different applications and later on also what are different analysis mode, because this is also very important in real-time PCR. It's not only doing the experiment, it's actually how you analyze the data that you get from the real-time PCR. So again, I made it as like an introductory webinar. And in this webinar, I'm going to follow it up with 2 different workshops. So workshop number 1 and workshop number 2, that we're going to discuss about analysis in much further details, because we cannot discuss everything today. And also the knowledge level among the participants and attendees is actually, is variable. So that's why I sent a survey. And in this survey, I was trying to explore what is the knowledge level of different attendees in terms of real-time PCR, in terms of a standard PCR and molecular biology, because that's very important and it helps me how deep I can go in this webinar. So I asked one of very simple question is that, have you ever used our RT-PCR method? And 38% have used it, and around 60%, and this was around 42 participants who answered the survey. So again, there are a lot of you have never used it. And then I actually asked a simple question about if anybody knows about standard PCR, like very simple molecular biology technique. So, and then 28% have experience with that, and 23% have medium knowledge, and 47%, which is higher, they actually don't know much about what is a PCR. So it actually gives me like a challenge of how I can use my webinar and how deep I can go in this webinar and what knowledge level I can go in this webinar to actually try to cover the knowledge level of all the attendees. So what I'm going to do now, for those who are expert in molecular biology, they might be a little bit at the beginning feels like a little bit boring, but hopefully it's not going to be boring. I'm trying to just to recap about PCR and real-time PCR. But slowly, I think after 10 slides, I'm going to go at the level that can be covered to everybody, inshallah. So try to start. Everything we're going to talk about today is basically on PCR, which is a polymerase chain reaction. And what is basically a PCR is just a molecular technique that you can use on nucleic acids, DNA, and also RNA. And it works on a double strand molecule. So you need to have like a 2 strand of molecule to work with the PCR. And you can use like nuclear DNA, like from the blood, from tissues. And people who works in the hospital, you can get even paraffin blocks from the pathology lab, and you can actually extract DNA. That DNA will not be fully intact, but still you can use a PCR on that DNA and it can work for you. People who comes from agriculture background, they can actually use the genomic DNA or can also use the chloroplast DNA. Another thing we can also work with is actually the mitochondrial DNA. And there is another thing called cell-free DNA. This is actually DNA which are not inside the cells. They are actually in your circulations. There is a lot of talk about it, and there are actually new techniques coming up in the diagnostic area, especially in cancer, where people use the cell-free DNA. This DNA can be in the circulation, inside the exosome or extracellular vesicles. You can also use the DNA from the viruses, from bacteria. It all depends on what type of application you are doing. If you are from the oil field, you can work on the bacteria that you get in the oil. If you work in the human field, like in the diagnostic in human diseases, you can actually extract DNA that might contain virus for you if you want to study like if there is a virus or no virus, or if there is a bacteria. And not only that, sometimes you do get a bacteria, like this is the bacteria in your blood. You want to know whether this bacteria is actually sensitive to a drug or resistant to a certain drug. Regardless of bacteria, parasite, any microbiological agent actually can get the DNA or RNA with that. And the only thing that you need here in PCR is just to design. Remember that in our DNA is a very long molecule. You cannot do PCR on the whole part of the DNA. So you need to select a specific region. If I want to select this region or maybe this part of the DNA, you need to have a tool to select this area. And our tools, actually, we use probes. And we sometimes call them primers. So you need 2 primers, one for each strand. So you need one for we call the forward, and then the other one reverse. So this is our selection tool. So among all the DNA molecule, I selected this region by just designing 2 primers that actually bind on the double strand molecule. And this makes the life easy. I mean, imagine that if you're using proteins, if you want to specifically bind to a protein, like in ELISA or Western blot or immunofluorescence, you need to have an antibody that is specifically bind to that protein. And antibodies are not easy to make, and they are very it's like, you know, they are more expensive. But here, in PCR, it's much simpler. It's basically a small tool that you can just design the primers and can bind to that specific region in your DNA. If you have an RNA molecule, because most of the RNA can be single strand, but we still have some viruses which are double strand RNA. PCR works on a double strand nucleic acid. So you need to convert this to a DNA molecule through reverse transcriptase. And we call it sometimes cDNA. In any way that this molecule, it needs to be a double-stranded nucleic acid. So you need to do an extra enzymatic reaction to convert RNA to double-strand DNA. So what I'm planning to do in one of the analysis workshops that we're going to cover is actually we try to design how you design primers. So this is something that you need to know. Primers are easy to make, but you need to know certain rules, and so you can get a better products. And remember that there are different applications of designing primers. So this is something that that you need to know about designing primer. That designing primer can vary between different applications. And in this webinar, I'm going to talk about application and try to cover certain rules. But again, this will be a topic of the coming workshop, inshallah. So this is the PCR. Now, how to do a PCR? If this is your double-strand DNA molecules, you need first step, you need to heat it up. You increase the temperature around 95, because with this temperature, the DNA, the hydrogen bond between the 2 strands will be broken, and then you're going to get the 2 strands separate. And then you need to add this 2 selection tool. We call them the primer. So primer forward, and then you have the primer, which is the reverse primer. You need to design them. There are software, you can do it manually, but nowadays softwares can do it much easier and faster for you. And then we call this step annealing step. So the primer, when it binds to the DNA, it's actually called the annealing step, where the binding step, where the primer binds to the DNA. And you actually lower the temperature around 60 degrees. Most of the primers, you can 50 to 60 degree primers can bind to the DNA. And then during this reaction, you're going to add the enzyme, which is called the Taq DNA polymerase. You're going to add the building blocks that makes the new nucleic acid, and you need buffer with magnesium. And this all comes in actually in a mix that you actually can buy and add to the reaction. And then you're going to do what is called the synthesis. In this synthesis, what's happened, the Taq polymerase is start to put, if there is an A here, it put the T nucleotide here, and then it keeps building it from the 3' end of the primer, and it keep going this direction. And then that's the upper strand, it's synthesized, and the lower strand is synthesized. So the end product you get from the polymerase chain reaction, that this area of the DNA, now after 1 cycle of PCR, like heating, annealing, and synthesis, you're going to get 2 copies. So this is, you're going to get 2 copies. And that's why after 1 cycle, 1 molecule of DNA, you're going to get 2 molecules of DNA if the efficiency is good. And usually in PCR, we do more than 1 cycle. So you go cycle number 1, cycle number 2, and usually can do 30 to 40 cycles. And usually in each cycle, you're going to get from 1 molecule, you're going to get 2 molecules. So that's why we have this as an efficiency is 2 to the power of n, where n is the number of cycles. So if you have 30 cycles, from one DNA molecule, you're going to get 1 billion copies of that specific region of the DNA. Remember that if this is our DNA, I specified the region by using 2 primers. So this region of the DNA, of having a one molecule, I'm going to get 1 billion copies. And usually in PCR, you don't start with one molecule. You start with like a bunch of molecules, like a large amount of molecules. So the amount of products you're going to get is very high. And that's why PCR is considered as a very sensitive method. And in viral, like in coronavirus, usually they use the PCR because you need only very little amount of that sample it can be detected. And this is all because amplification in PCR can make you thousands of copies, like billions of copies you can detect them using different techniques. So all of these products, you can use them different technique to detect them at the end of the cycle. Now, something I want to mention here, which I'm going to talk in the application later on, is that if you start with more material, for example, I'm using one molecule, after one cycle, I'm going to get 2 products. But if I use 10 molecules as a starting, after one cycle, I'm going to get 20 molecules of products. So that's why the more starting material you have, the more products per cycle you're going to get. And why I'm mentioning this is important, because one of the applications we want to quantify, and this is actually the basis of quantification. The more starting material, the more product you get per unit time. So this equation that I'm showing you here is based if you have one molecule. If you have 10 molecules, say 10 times 2 to the power of n. So you're going to get more every cycle. You have more products per cycle. If you start with more, with the starting material is much more. So before we go for the real time, I just want to mention for people who use polymerase chain reaction. Remember, I told you that in the PCR, we actually do heating, then we anneal with the primer, and then we do synthesis. If this is the axis, y-axis is the temperature. If the y-axis is the temperature, and this is the time in like a minute, you see that in PCR, we do one cycle of heating, and then we anneal the primer, and then we do the synthesis. And then you keep doing it 30 cycles, one by one. If I go back to the previous this slide, as I said, that you're going to do heating, and then you anneal, and you do synthesis. This is one cycle, and you have to repeat it many cycles, around 30 to 40. And this is actually, you need a machine that we call the thermocycler. From the name, is that it cycles the temperature. So this machine, you put your sample tubes in the plates, and this tubes or plates, you can put them here, and you operate the machine. But at the end of the day, you need to take this tube out. And then if you want to collect the data, you need to run another experiment. And some people use an agarose, and then you can also, after the agarose, you need to have a camera to detect the products. So if this is a PCR product that's been formed because of amplifications, and this requires a lot of time. It is labor intensive and also time consuming, and it actually it can be painful for the people who are working in the lab if you have so many samples like 100 and 200. In some of the PCR technique, you need to use enzyme. So you have to get the tube, you add an enzyme, and then you run it on a gel. Some of them you need to have different specific primers. Sometimes you need to heat the product. So different actually modification of the PCR for different purposes that I'm not going to discuss in this webinar. But what I'm trying to tell you that if you want to collect data on the PCR here, it's not only doing the PCR like thermocycling, you need to do extra experiment, and this actually will take time. So instead of getting data in one single day, you can get it like after 2 days, and also the lab technician will be very pissed off because if you give him like 100 samples, if you have ordinary PCR. So that's why people actually invented what is called a real-time PCR. And in real-time PCR, it is again a PCR where you need a thermocycler that actually changes the temperature. Remember, this is heating, 90, and then you anneal the primer, and then you do DNA synthesis. And this is one cycle, and you keep repeating it one by one. But in this method, in real-time PCR, and this is actually the slide I'm trying to describe what is a real-time PCR, you're going to collect the data while the PCR is being run. So I don't have to wait to get the sample and do agarose gel, or I'm going to do detection with the camera. Here, basically, this machine will be modified in such a way that you're going to get the data on your PCR on real time. So that's why the name real-time PCR. And this is actually achieved by actually adding some molecules in your PCR. So if this is your PCR tube, you have the DNA, the primers, and the tag polymerase, you're going to add extra material to this tube. And this extra material actually can be actually affected by the PCR. So you can collect data, and most of this material actually are fluorescent molecules. So basically they can shine colors. So you need a light source, and this is actually a lamp, or it could be a laser. I don't think most of the real-time PCR, they are not equipped with laser, they're equipped with the lamp. And this lamp actually excites this molecule, which is in your PCR tube, and it will shine a color that you can measure with the charge-coupled device camera. And this can measure the intensity of this fluorescent molecule during this PCR cycle. So basically what it means that during the PCR, while the PCR is run in the machine, you're gonna collect data, and you can tell the machine, for example, I put the camera here, it's basically I'm telling the machine I want to collect the data when the PCR ends, every cycle. So every cycle, it will measure the intensity of fluorescence intensity of this molecule at the end of each cycle. And by this, you're gonna get data on real time. Now, however, I'm gonna go in the webinar trying to discuss how this is actually being utilized and how this is gonna help you to answer certain questions in molecular biology. So this machine is being modified, and it actually looks different because you need to measure fluorescence. And this slide, I'm just trying to describe to you people who use fluorescence in flow cytometry or in immunofluorescence or even in ELISA, that you need to excite it with the lamp, with the light, and that molecule will change, it will absorb, and it actually emit a new wavelength, less energy, and this is called a Stokes shift. And that's why each fluorescence molecule has an emission wavelength, and this wavelength is very important because different molecules emit different lights. And with this, it actually helps us to do multiplexing that I'm going to discuss in the coming slide. So this is actually what it looks like in a real-time PCR machine. It's basically a thermocycler. Basically, this is the block where you can keep the plate. This is actually your 96 well plate. Again, you can also use 384 well plates if you have more samples. And then you're gonna get a light source that actually hits the sample. And then the sample will emit a new light, which goes to a CCD charge-coupled device that can measure the fluorescence intensity of each well. This is actually a snapshot of the plate in the machine. You can see each well will emit a new light, and this light can be actually measured by the camera every cycle. So after each cycle, you're gonna measure the fluorescence of each well one by one. Now, that's why the machine will have, it's not simple. It will have a light source, and it will have a CCD camera and a detector and a cooling, because you need, this camera can become hot, so you need a cooling system for this camera. So that's why the price of this machine goes up, because of this setup, engineering setup. Now, there are other machines, actually we call them the rotor-based. Instead of having like the whole plate, you take the measurement of each well in one snapshot. This requires a lot of calibrations and also alignment with the camera. The rotor machine, it has less calibration. In the rotor machine, basically the tube are actually arranged in a rotor. So this is how you arrange the tubes. And then you have one light source here that hits one tube at a time. And whatever this tube will emit light, the CCD camera will detect the intensity. So what's happening, this rotor will keep rotating. So all the tubes will be measured by one light source and one emission, one filter. And this is all, will give you much more reliable data than the other one. But both machine works actually. So in the practice, if you go to the practice, this machine actually the one that is plate-based is commonly used, and also the rotor machine are also commonly used. And they both give you good data and reliable data. But again, in terms of efficiency, people say the rotor system give you better efficiency. But again, this all depends on your budget, how much money you have to buy an instrument in the lab. And very common machine, you can see actually the applied biosystem machine, actually now Thermo Scientific have it. Is a very common, you can see it in many labs. And you also have a machine like Roche used in many diagnostic lab and also research lab. But again, there are some, it's called cube rotor machine, which are actually, you can hold it in your hand. And this is like very cheap. You can get it, if you have like a small lab, you can buy like the cube RT-PCR machine. which is basically a cubic machine that you can actually lift it with your hand. It's not a very heavy machine, but it has limited application. So if you have less budget, you can buy the rotor machine from the market. Now, so that's basically what I try to describe for you. Is basically what is a real-time PCR. It's basically a machine that can measure data during the PCR. So that's as simple as it, without going into complications, but it's just for you to, at this level, real-time PCR is a machine that actually measured PCR data in a real time while the PCR is being run. And I showed you that the plate system and the rotor system. Now, let's talk a little bit about these fluors, fluorophores, the molecule that you add to your PCR. If this is your PCR tube, you're going to add your primers, your template DNA, and you're going to add the Taq polymerase, but then you need to add a molecule here. And this molecule, either two of them, actually, we classify into two groups. One of them is the cybergreen, which is very commonly used, or the other group, we call them fluorophores. There are many, and they have an extra molecule called the quenchers. And I'm going to tell you why the cybergreen does not need a quencher, but this one needs a quencher. And we're going to define what is a quencher, what is a fluorophore in the coming slides, inshallah. Now, cybergreen is actually a molecule that's been patented. I think it lost the patent in 2016, so no more. You can make it if you have a very good synthetic chemist. You can make it in your lab. But this is actually a molecule that was patented for a long time because it actually had a lot of business, too much money the company got from it. And the company had this is the Life Technology actually got the patent for this cybergreen. Now, what is this cybergreen? It's actually a molecule that does not bind to a DNA when it is like a single strand or double strand. It only binds to it whenever the double strand is being formed. So if this is a double strand molecule and this is single, whenever this is actually binding to the other strand, this cybergreen start to bind between the bases in the intercalate between the two bases in the two strands. So it preferentially binds to double strand DNA. Now, what does it mean? So remember that in the PCR, when I have a PCR, this is my PCR, I'm going to put the two primer number 1 and primer number 2. At the start of the reaction, the fluorescence in this tube will be very low because cybergreen is there, but it does not fluoresce. It only fluoresces whenever you form a new double strand molecule, it starts to bind to the DNA. So whenever this is like the DNA is being synthesized, it comes and intercalate between the bases. So after each cycle, you get more fluorescence. So that's why it preferentially binds to double strand DNA. So if you have, this is my DNA. Let me just change the color of this erase and then color of my pen. Let's see, this is my DNA and this is my primer and this is the Taq polymerase. So during the synthesis, when you form a double strand molecule, the cybergreen start to bind in between these bases. So after the end of the cycle, so remember this is the cycle. During the synthesis, I put the camera and I tell the machine only measure the fluorescence when the cycle ends, before you heat it again. So that's why after one cycle, you're gonna get cybergreen molecules in this new 2 double strand molecule. And then if I have my Y axis and X axis here, and I have here the fluorescence intensity, and here I have the cycle. After one cycle. So after one cycle, I suppose the fluorescence of the tube will go up because now I'm getting 2 products that will have cybergreen. Now, second cycle, I'm going to get 4 products if I'm starting with one molecule. So again, the fluorescence will keep going on. So after each cycle, the camera will start to measure the fluorescence because the cybergreen only binds when you form a new double strand molecule. So that's why continuously you get increased fluorescence. Theoretically, fluorescence should go linear because this is a linear when you have 2n, معناته 2, it means that you're going to get 2 products after each cycle. And this is when the efficiency is 100%, that after each cycle, I'm going to get 2 copies. So that's why after each cycle, you're going to get more copies, more cybergreen bound, and more fluorescence in the PCR. But in reality, this curve will not look as a line, as a linear line. It will look like an S shape. And why is that? Because at the beginning of the PCR, you're going to get very few molecules. There is a fluorescence, but the camera that you have, the detector, is not sensitive. It cannot measure fluorescence at very low level. So that's why after a few 5 cycle, 10 cycle, you're going to get it like the same fluorescence in the y-axis because the camera is not sensitive. But there is a time when you get enough product, the fluorescence will go into an exponential phase. And then after, when you have too much fluorescence, because after like 25 cycles here, the tube will be fluorescence like a green color. For example, if you're measuring green light, it will be too green that the machine cannot tell the difference. So that's why you're gonna get a plateau. So that's why in a PCR, you're gonna get a sigmoidal curve like this. And the beginning of the curve is because the camera is not sensitive to look at the difference in fluorescence. And the end of the curve because of the fluorescence saturation. And this is a problem you get in flow cytometry, in ELISA, that when you have too much fluorescence, the machine or the detector that you have in your plate reader or in your fluorescence detector cannot tell the difference. So that's why we have this S curve. And usually we measure the fluorescence, the detection here, in the data analysis, we look at the exponential phase of the PCR. So that's what basically is the cyber green. It's the first molecule that we want to talk about. Actually, it's the molecule, as again, to repeat, is a molecule that actually during the PCR, whenever you form a double strand molecule, it will intercalate between the two strands in here and here. And every cycle, you're going to get more fluorescence because you're going to get more PCR products. And again, this cyber green it's not specific for a specific sequence. It only binds randomly to any double strand DNA that's being formed. So every cycle, it does not basically, so why I'm telling this, because I'm going to mention later on that you cannot use cyber green to multiplex because it cannot identify a specific sequence. It basically binds to any double strand DNA that's being formed in the reaction. Now, the other format of real-time PCR, the molecules that we use, is called the fluorescent dyes. And there are many dyes. Roche has different, and the applied biosystem have different. And people are still making new one. But the common one actually are this one. So cyber green is one of the dye. There's one thing called FAM, there's called VIC, Joe, and Tamara, Ned, and ROX. They are different molecule, different structure that you add to your PCR tube. But this molecule, unlike the cyber green, they fluoresce all the time. So all the time they are fluorescent. It's not like cyber green, it only fluoresces when you make the PCR double strand, it will fluoresce later on. These molecules, if you put them in their reaction, it will always be shining color. So during PCR, if I'm doing my fluorescence and cycle here, cycle number, I'm doing fluorescence. So they will always be high and will be same levels during each cycle. So what we do here in this fluorescence, you need to add an extra molecule we call the quencher. So this is the quencher. What is the use of the quencher is that whenever you make the PCR, this quencher is close to the fluorescence molecule. So that's why this molecule will have very low intensity at the start of the PCR. So if this is the start of the PCR, it will have very low fluorescence because this molecule is very close to a molecule called quencher. And this is actually something called FRET in physics, fluorescence resonance energy transfer, because this will not fluoresce because this is a molecule that actually inhibits the fluorescence of this molecule. The only time it fluoresces when this quencher is kept away from the molecule. So how do you do in a real-time PCR, how we actually use this is actually you design what is called a probe. And a probe is basically a single strand DNA molecule. And then it has a fluorescence at one end, the molecule. And this molecule could be VIC and FAM or TAMR or NET. And each one of them will have a quencher. So this probe, you need to buy it. You need to design, you need to buy it. And it actually makes if you use this one, it's more expensive than cyber green because of this arrangement. And what you do here, you design your primers. If this is your PCR, you design, like the cyber green, you design forward and reverse primer. But in one of them, you're going to put an extra piece of DNA, it's called the probe, and it only in one of the strands, and it will have a fluorescence and a quencher at one end. So what happened during PCR, and just trying to, before I go there, that because each molecule has different filter and different wavelength, you can multiplex here. Unlike the cyber green, you can multiplex these dyes. So this is actually what's happening in a PCR. So again, you need the tag polymerase, you need the DNTP and the buffer like cybergreen. But unlike the cybergreen, you need to add an extra molecule we call the probe. And this probe will have a fluorescence molecule attached at 5' end, and the 3' end, you have the quencher molecule. So at the start of the PCR, this tube will not have fluorescence, or it has fluorescence, but very low, like very low fluorescence. But during the PCR, the tag polymerase will bind to the 3' end and start to move in this direction. And whenever it comes here, it will hit this probe. Tag polymerase has an activity called 5' nuclease. Whenever it sees something in front of it, it starts to break it. So when it breaks this probe, the fluorescence molecule will be away from the quencher and start to fluoresce. So this tube, after one cycle, if you have this, again, the curve, if I'm measuring the fluorescence of Vic, one of the dye, which is the green color, for example, and this is the cycle number, after one cycle, I'm going to get one fluorescence molecule being released. So it goes up. And then 2 cycles, it keeps going on, it keeps going on. It appears to be linear. But again, because of the CCD camera and because of the fluorescence saturation, you're going to get an S-curve like the cybergreen. So again, if you're using a fluorescence molecule, you tell the camera of the machine that at the end of the cycle, I want to measure the fluorescence of this fluorescence molecule. For example, this is the Vic. And because after each PCR reaction, the tag polymerase is going to break one probe molecule. So you're going to get one fluorescence molecule released. So here, after one PCR reaction, you're going to get one fluorescence molecule. And here you're going to get 2 fluorescence molecules. So after each cycle, the fluorescence should go up one by one. And again, you're going to get a sigmoid curve like the cybergreen. But the only difference here is that you're going to use a probe. And this probe specifically binds to the DNA, and you can multiplex that. So let's say that we talk about multiplexing here. So if this is like somebody of you like want to measure, you have a sample from the plant or sample from an animal or blood, and you want to measure 4 strains of bacteria. And what you need to do, you need to design a primer, each one of them, reverse and forward. Specifically for each strain, you need to specifically design primers, one by one. And then if you're using cybergreen, remember cybergreen does not differentiate between sequences. So it binds to any PCR product. So all the fluorescence you get in the tube during the PCR, it will come from all the strains. So that's why cybergreen cannot differentiate between them. The only time cybergreen can differentiate when the size, PCR product size is different, and I'm going to talk about it in one of the applications, but not now. But if you use a fluorescence dye, like a TACMAN, or sorry, the fluorescence probe that I mentioned to you later, what you can do here, you can design a probe, specifically binds to each strain. You give this one like a red color, like for example, Psi5, and then you can have like this one, a blue color, different wavelength. And this, you design another probe. Each probe will have a quencher, a specific quencher for that one. And this is, for example, green. And then this is like a lighter yellow color, UI. So you design a probe for each strain, you need 2 primers, and you need a probe specifically bind to strain A. So during the PCR, the machine, because it has different filter for each of the color, you can actually measure all the products in one single tube, and it gives you an answer. And this is actually what you do in coronavirus. In coronavirus, usually we have like multiple genes. We don't measure one gene. And then if you want to be positive, so that's why we actually try to mix 2 colors in one well. So this will save time and also save money. Instead of running each strain in 4 tubes, I can run them in 1 tube and I can get the answer in one single reaction, one single day, and you can get the answer immediately. So that's basically what it means for multiplexing. Now, just for your knowledge is that, remember in real-time PCR, we basically use fluorescence molecule. But the fluorescence, the main problem of fluorescence is that you cannot multiplex many color. Because something in fluorescence you should know is the overlap between the different wavelengths. If these 2 wavelengths are next to each other, then it's become very difficult to differentiate, for example, the green to a light green or green and yellow. So that's why you are limited with how many multiple. So you can, in the current mode in RT-PCR, I think you can with the, I think, applied by system, they generated new dyes called GAN and ABY. These are two different dyes. They actually made a new one. ABY and GAN, these are two new dyes. And there is something called Mustang Purple. And you can mix VIC and FAM. So the current version of, if you want to multiplex, I think you can only get 5 products. They can multiplex 5 genes in one tube. And that's a problem with fluorescence because and fluorescence saturation, and also because of multiplexing, you are limited with that. So there is a new modification where people use mass spectrometry. In mass spectrometry, it's different than fluorescence, that you can multiplex as much as you can. And the new format, you can actually do it in a real time. And this machine is called the sequenome or mass array, or we call epityper. It's actually a MALDI-TOF, matrix-assisted laser desorption ionization. This is ionization and time of flight. This is one of the methods in mass spectrometry that you can also use it for protein, and that's called biotyper. People use it to identify bacteria, different strain of bacteria. But if you don't want to use it for protein and you run DNA molecules, then it's called epityper. It's the same machine. You only need just to change the software that can tell you the analysis. But this is, I'm not going to discuss today. This is a totally different method. But again, here it will become an advantage. If you want to measure in one single tube, you can go up to like 30 to 40 multiplexing in the tube. And people who studied genomics, like SNPs, multiple SNPs, they can actually run this plate and you can get many data from one single tube. And also the probes for this are cheaper compared to the fluorescent molecule. So again, this will give you a cheap result and much more multiplexing if you want to use mass array, mass spectrometry for detection. So here I'm trying to, what we discussed so far. I told you about what is the PCR. We told the PCR is a method that you actually need 2 primers, and you can amplify a specific region in your DNA. And then we discussed real-time PCR. That is basically a PCR, but you get the data from on a real time. And then I discussed that we use 2 types of fluorescence molecule. We use cyber green, which does not fluoresce. It only fluoresces when it double strand is formed. Or you can use different fluorescence, but you need to have a quencher with that. So this is basically was an introduction about what is real-time PCR, people who don't know what is PCR and real-time PCR. And now I'm going to move out with different application and analysis. But before I want to go there, I want to see if anybody has questions, because you can ask question in the chat. I think we have the chat here, question and answers. I don't see any questions, but if you have any questions, you can ask before I move to the topics, which gets a little bit more advanced. So let's see, I have around 130 participants. So Yusuf, can you please show me if there is any questions? I don't see here.
Yes, Doctor. If anyone have question, you can ask now, please.
Yeah. Okay. Okay. Okay, so I have one question. It says, how does the probe binds? The probe binds like the primers. So you only need to change the temperature. So the temperature, whenever you like binding the RT-PCR, so if this is my DNA, which is denature, I'm going to change the temperature for the primers to bind. The same idea with the probe bind. So you need to just control the temperature, and that's from that one, that's the way how the probe binds to the DNA. Now I got another question, is it quantitative? Yes, actually I'm gonna talk about one of the application if you wait a little bit. So we're gonna discuss about how PCR can be used to quantify, okay? So I got 2 questions only. So anyhow, so I'm gonna continue now. So what I want you to remind you, please, you need to, if you have any question during the presentation talk, you can please ask them now. I'm going to have another question and answer session at the end of the talk, because remember that this is an introductory talk about it, okay? Now, I got another question is that what type of statistical analysis? Okay, so I'm going to talk about analysis. Remember, as I told you, the first part is just to introduce you about RT-PCR. And then I'm going to talk about different applications of detection, genotyping, because each one of these will have different analysis. So that's why if you become patient, I'm going to talk about different analysis, inshallah. Is the temperature effect on quality of sample? I didn't get that question, but if you're talking about sample quality, remember that the DNA is more stable than RNA. Usually with DNA, sometimes if you have a very good extraction method and clean extraction method, you can keep the DNA room temperature and you can ship it within a week if you want to send it somewhere else. So that's not a problem. You have a very good experience with we do get samples from Kuwait, from Iran, from Iraq, because sometimes we do some work with sample and we actually ask them to send them on the room temperature. And this is provided the DNA is being extracted. Now, however, with RNA is not very stable, and we have problems if people like shipping RNA for us, or we also sometimes ship RNA. RNA needs to be frozen, and that will help in the quality. Some people add some RNase inhibitors, so to keep the RNA on room temperature longer. So if you work with patients, like if you're working in the field from animals, you're collecting blood, and this is an RNA experiment, I advise you that during the blood collection or tissue collection, you need to add an RNase inhibitor in there. And that will help actually to keep the RNA in +4, like if you put it on ice before you bring it to the freezer, that actually help with that. All right, so now what I will do, okay, can we sequence an RT-PCR products? Yes, actually what's happened after the PCR, you do the RT-PCR method, you're gonna get a PCR product. Now that PCR product, there are ways actually, there are ways to actually sequence those. So, and if you are worried about the fluorescence intensity, that's not an issue because remember if you're using the cyber green, you can actually try to, you can purify that product and you can run it for sequencing reaction. So you don't have an issue. You can sometime, but you need, as I said, you need to make sure that the data. Now, again, if you want to ask about sequencing, remember sequencing is a different technique. We use what's called the Sanger sequencing or use the next generation sequencing. They are totally different technique and they can be used to sequence. Here we are not sequencing, we are actually detecting, genotyping and quantification. And most of the people want to study quantification of the PCR. All right, so now I would like to move up with the talk and we try to discuss more. So,
Doctor, please. There is a question in QA.
QA, sorry, open question. Okay, so somebody asked me about CT value. Okay, I'm gonna talk about CT value when I'm talking about quantification. So Iman asked about that question. Are any rapid tests less than 40 minutes? Yes, Abdul Rahman asked about if I'm gonna go back in one of the slides. Remember I told you that this is the PCR cycle. If this is your data fluorescence and this is your cycle number, remember that we are detecting fluorescence after each cycle. So one of the way to make the PCR much faster is actually you lower the cycle time. And this is if I go back here, this is like, for example, this is the time. This is actually, it takes about from here, one cycle to here, it takes around 5 minutes, for example. So now there are some kits, if you want to make this go faster, there are actually kits with tag polymerase that this can be done in a much faster way. So you can get a PCR result less than 40 minutes. Actually 40 minutes, 30 minutes, 40 minutes, you can get a data from 96 samples. So yes, if you say any rapid tests, yes, there are rapid tests. But again, the only difference you need to use is actually the master mix. This is the enzyme and the buffer you're using. It becomes much more expensive. Usually when RT-PCR, it usually takes around 1 hour and 30 minutes. Depends on the application. Some application can go longer than that, around 2 hours. So, but if you use a rapid test, you can get the data in half an hour, actually, half to 40 minutes. All what you need to do is just to lower the cycle number, because remember, this cycle is repeated 30 times. So if you have 5 minutes, 30 times 5 is like 150 minutes. But if you have like 1 minute cycle, then you need only 30 minutes to get the data. So again, yes, you can do that, inshallah. So let's continue. I'm going to talk about here. So I have a lot of questions. Okay, so let me erase them. Yeah. So what I'm trying to come back again, remember that for people who came late, I actually asked the questions at the beginning. How many people knows about PCR and how many of the attendees knows about real-time PCR? And we have a large number of attendees who, for them, this is like the first time you hear about RT-PCR. So that's why the first part I spent mainly about describing what is PCR and what is an RT-PCR, and the two methods, the cybergreen and the fluorescence probes. And why I mentioned that? Because it will help the participant to know about different applications. Now, as I said, this is an introductory seminar, a webinar, where basically I'm going to talk about detection, about different analysis, but this will be followed with two analysis workshops. We are not going to discuss analysis in detail today. And I said, one of the workshops I mentioned is actually how to design primers and how to design multiplexing for different applications like genotyping and detection. Now, the second workshop, I'm going to actually spend good time actually on how you quantify. And many people actually heard about a very common quantification, but I can tell you that whenever you want to submit a good work and you want to submit something that actually reliable, you need to know about different formats of analysis that I'm going to discuss in the coming slides, inshallah. Okay, so let's discuss first about the detection method. Now, in detection, there is only two questions you need to answer. For example, I have one of you once, like he got a meat in the market, a court case. And the guy said this meat is actually a cow meat, a lamb or a cow. But the court said, no, this is a meat from a gazelle, because the guy was caught, he's hunting gazelle, and you want to understand whether this is actually a gazelle or a cow. So you need to run an RT-PCR method. You want to say, answer yes or no. Is this a cow or is it a gazelle? That's one of the questions. Now, another one is actually, you have a patient who have a mutation. You want to answer, does this patient has the mutation, yes or no? So that's the answer I want. I'm not interested in the quantity. I just want to say yes or no, absence or presence. Now, another question you also need to get, for example, infection. I have a patient who has an infection. I want to know whether this patient has a TB or no TB, like tuberculosis. Or for example, the current problem is the corona, whether this patient has coronavirus or no coronavirus. I'm not interested whether he has too much virus or less virus. I'm not interested in quantity. I want to do a qualitative analysis, whether this guy is present or absent. The other thing you need to know is like strains, as I told you, is this strain present or not present? If there is a bacteria, does this bacteria is resistant to certain drug, yes or no? So that's basically what you need to do in this application, is basically you want to say yes or no in the reaction. So what you do in this protocol, you can use the cybergreen fluorescent dye. All what you need to do, you just design two specific primers, like an ordinary PCR. And this primer will give you the answer yes or no, if you get a PCR product or you don't get a PCR product. And then you do the fluorescence, and then you can see here, if this sample, this primer does not bind, you're not going to get a PCR product, so you don't get an amplification curve. So this sample is all negative. Now, if the primer binds and you get the PCR product, you're going to get this amplification curve. So all this sample will have yes. So you can run 96 sample in this well, and it can give you the answer of 96 sample, whether yes or no. You can also do, if you have the PCR block of 384, you can run 384 sample in one single 30 minutes, 40 minutes, you're going to get the answer. So that's how fast it is. Or you can go the other way. If you want to get like more expensive, if you want to be a fancy person, you have too much money, you can go for a probe-based. So again, you design the same primers, left and right, but you're going to design a probe that specifically binds and that gives you the answer. And this probe it's gonna actually, as I mentioned, it's gonna go broke down. Whenever the fluorescence increase, معناته إنه yes, this is a positive sample. If there is no fluorescence increase, you say no. And you can easily tell from this curve that this is a sample. And you can actually tell the software by looking at certain CT value. I'm going to talk about CT value later on. That if this is CT above 24 is positive, below 24 is negative. So you can tell the machine, and the machine can give you a report immediately within 40 minutes. So that's one of the application. Now let's say give you one example. So try to understand what I'm trying to mean here. Suppose that you have a sample from the field. It could be a plant with the disease or patient with the disease. And this sample, you want to see whether you're going to get the whole DNA, and then there is a DNA of that specific bacteria. So you design a primer that only binds to the bacteria, and you run CyberGreen. If you see here, then you say, yes, this sample is contaminated with that bacteria. Okay, I got another person, he wants to know, okay, the bacteria is present, is there. I want to know if this bacteria, does it have a specific mutation? Like, you know, that, for example, let's mention about the MRSA, methicillin-resistant Staph aureus. So this is a Staph aureus, a bacteria. I'm going to design a probes and a primer. I can design a probe that is a green color to tell me that actually this is a Staph aureus. But I know that in this bacteria, remember this is like a bacteria DNA, circular DNA. I'm going to design two primers that tell me that this is a Staph aureus. And there is a mutation that actually makes this Staph aureus resistant to certain antibiotic. So I'm going to design another two primers and then another probe with the red color. And then I can multiplex them. I can put the 4 primers with the 2 probes in the same tube. And then my machine will gonna tell me, whenever I see, remember that the Staph aureus is a green, I see green color, معناته yes, there is a Staph aureus. But if I see also green and red together, it will give me the 2 answers together. It says that there is a Staph aureus, and also this Staph aureus is resistant to that antibiotic. So this is one of the application people use, and actually we use the same technique for coronavirus. In coronavirus, we actually have one gene that tell me that, okay, this is a coronavirus. Remember, there are 5 coronavirus that can infect human, not only one. The 3 severe SARS-CoV-2, which is the COVID-19, and the MERS, the Middle East coronavirus. So we run one gene that can tell me, yes, this is a coronavirus. And then I run another 2 genes in the same well, okay? It can tell me, okay, this is a coronavirus of SARS-CoV-2. So this is how people actually design. So again, you need a lot of work to design primers and probe. So that's why I'm gonna have another workshop for you if anybody is interested later on, how we design primers and how you design probes. Not only that, if you are interested in multiplexing, I'm gonna actually talk about fluorescence and fluorescence overlap. Remember, there is something called FMO, fluorescence -1. There's an analysis you need to do that we're going to discuss here and how it will help you to multiplex. And then the other thing you need to do is how you control your extraction method. Now, why I'm doing that, remember that if you have a DNA sample, if this is my DNA sample, I'm going to do a real-time PCR. And then I'm going to get these results, the one I see in the graph. Sometimes I don't have DNA, I have an RNA. This RNA, I have to do reverse transcription to get a DNA and then PCR, and I'm going to get the result yes or no. But in some fields, especially like in corona, when you have a disease, it's very important. Sometimes you're going to get this negative reaction, means the patient, it has 2 meanings. Either a patient does not have the virus or does not have the bacteria, or maybe your PCR and your reverse transcription did not work perfectly. And this is something which is dangerous if you have a patient who have the disease, but you tell him that he does not have the disease from the RT-PCR. So what people also do during this reaction, they do what is called an extraction control or a control reaction for this. And all it does is actually, because whenever you have a samples, this is my sample that I got from the patient, I'm going to get the RNA from the virus, but also I'm going to get RNA from the human. Let me see here, pen color, blue color. And get too much RNA from the human RNA. But there is only one RNA molecule, for example, it has only one virus. I'm going to get one RNA molecule from the virus. So what you do in this assay, you also need to design primers that actually target a human RNA molecule. So if the human RNA molecule is working and the viral RNA molecule is not working, then you can assure that this patient is a negative patient. Because if the human RNA sample target is working, means your extraction was good and your reverse transcription was good and your PCR was working. So this will give you an assurance. So that's what we're going to discuss in this workshop. I'm going to discuss multiple things. Number one, how you specifically design good primers for different methods and how you design probes. If you are interested to run more than one sample in a tube, I'm going to discuss this, multiplexing, and how you control for an extra one by looking at different extraction controls. And actually, we are currently now designing some assays, and we are actually using a mouse sample on a human sample. So these are different ways you can do that, inshallah. Now, the second application is called the genotyping. Now, people who don't know what's genotyping, basically, I'll show you, for example, here, I have like around how many guys here? 1, 2, 3, 4, 5, 6, 7, 8 people here. And then in this 8 people, and this is a sequence in the DNA. In this location, in this locus, we call it locus, okay? In this locus, there's also G. All of them have G. So we don't say there is no variation. So all the human, all these 8 people, all this population will have a G in this location. Again, A also same and T same. But if I go on the 4th position, on this 4th locus, I can see some people having an A and some people having a G. And this is why we call it a single nucleotide polymorphism. Polymorphism means different shape. And that's why we call it A to G. So some of you, if I take of the participants, some of you will have an A, some of you have a G. So we call it SNP. So in genotyping, I want to know if I have a people of 100, like 1,000 people. I have one sample of 1,000 individuals. I want to know, using real-time PCR, whether this individual is an AA, because remember that we are diploid, we get one chromosome, one DNA molecule from the father and one from the mother. Or it could be an AG, you're heterozygous, or it can be a GG. And this is what we call the genotyping. Genotyping means I want to know whether you are an AA, you are AG, or a GG. For bacteria, it's different, but in human, we are diploid. Remember, diploids mean you have 2 DNA molecules for chromosome number 1, for example. We have chromosome number 1 coming from the father and coming from the mother. If you are a homozygous AA, you're going to get A from the father and the A from the mother. We call this alleles. This is the different forms of the sequence. We call them alleles. So you have 2 alleles, one from father, one from mother. So you are an AA. Now, if you have A from the father and G from the mother, then we call you a heterozygous individual. Now, the question here, how we can use real-time PCR to get an answer for this? And this is actually, we use, you can use CyberGreen, there is a way, I'm gonna talk about it, but mainly use what is called a probe technique. So again, this is an individual who's an AA. It means that he has the A from the father and A from the mother. Remember, our DNA is double strand. So A will bind to a T. Again, if you're a homozygous GG, you're gonna get G from father and G from mother. If you're heterozygous, you're gonna get A from one of the parents and G from the other parents. So how to answer this using a real-time PCR? What you need to do, if this is the location, you need to design a forward primer and a reverse primer. So you're gonna have a tube. You put the DNA sample, which is of that individual, and then you're gonna put a forward primer and a reverse primer. And then you're gonna add 2 probes, 1 probe for each of the alleles. So this is a probe that actually have a Vic fluorescence. Let's say that's actually Vic is in the green line, so I call it blue color. And then you have another probe, which is the C, which it binds to the G. So this probe, what is the idea? It basically binds completely to this sequence. And these probes will bind to the G allele, and then it has a different color. So you need to add 2 probe for A and probe for G. The only difference is they have different color, blue and FAM. And each one of them will quencher. So at the start of the PCR, they have very low fluorescence. So they are not fluorescent. Now what you do here, so this is why in each one we'll have 2 PCR primer and 2 probes. So for each individual, for each PCR tube. So what's happened during PCR, you take 2 measurements. You take before the PCR for each of the tube. If this is an individual has an AA, this is individual the GG, and this is a different PCR tube. Then after the PCR, because the tag polymerase, if I go back here, during the PCR, the tag polymerase, if this probe binds to this one, the A1 binds, then this reporter, which is the fluorescent marker, will be separated from the quencher and start to fluoresce. So what's happened if you have an individual who is an AA? If you have an AA, means you have 2 chromosomes having A, one from father, one from mother. And this probe will bind. This probe will not be bound because it has like a mismatch in the center, it will not bind. So this probe will be bound. So during the PCR, the tag polymerase only breaks the Vic from the quencher. So that's why after PCR, that tube will shine only blue color. So I can say that this individual is an AA. Now, if you have an individual is a GG, again, you're gonna have 2 GG, 1 G here and 1 G here. So which probe will bind? This will not bind. The FAM will bind, which is fluoresce in the red color. So after the end of the PCR, if I take the fluorescence, it will only shine red color fluorescence. So I can tell that this individual is a homozygous for GG. Now, the third case, I have an individual will have 50% is an A, because we remember we get 1 chromosome from father, 1 chromosome from the mother. So 50% will be A, the other 50 will be G. So in this case, the 2 probe will be bound, the VIC and the FAM. So this 2 probe will be bound because you have the 2 templates is there. So after the PCR, you're gonna get 2 fluorescence, the red and the blue together, and you can tell me that this individual is a heterozygous. So sometimes you see in the y-axis called delta Rn, you subtract the post-PCR intensity minus the pre-PCR intensity. So that's why in this technique, you're gonna get a scatter plot where the x-axis is the intensity of the A probe. Remember the A probe is the blue color, okay? And then you get the intensity, which is 0 to maximum for the G probe, which is the red color. Now, individual who have very low blue color and very high red color, they are the GG. Remember that I'm talking about the plate here, 96 well. So these are 96 samples you run, and the software will actually scatter them. If you have a sample with very high fluorescence blue and low fluorescence of the green, then this will be the AA. The one in the middle are the heterozygous. So very fast, you can actually genotype an individual by using a real-time PCR. Now, how you use this, if you want to detect if the person has a G6PD, or for example, if this person has sickle cell disease, if you want to diagnose a certain mutation, like a very common, like beta thalassemia, alpha thalassemia, there are some mutations which are common in Oman. You can design probes for those mutations, and you can actually get the answer very quick in 40 minutes. All what you need to do is just to prepare a master mix with the probes and the primer, and you put them in a 96 well plate and run you load each sample in each well and put it in the machine. Within 40 minutes to 1 hour, you're gonna get the data of 96 individuals very carefully. These are actually the negative control. So the sample without, so you should be very low. So whenever you want to ask what are these are the negative control. So that's basically one of the application that we're gonna use we're using the probes. But if somebody said, okay, Doctor, I have the probes are expensive. Yeah, because probes actually, I mean, if I want to say the price in Oman are more than outside, I think it costs around 150 Omani Riyal if you want to buy a probe. But remember that this probe, it can be run for thousands of samples. So if you have like a project that actually run many samples, it becomes cheap for you. Now somebody said, okay, can I use a cyber green, like to tell me what is the genotype? The answer is yes, and this is actually what we call the high resolution melting, HRM method. In HRM method, it's actually much cheaper than the TACMAN probe, which is the allele discrimination, which I described in using a probe. But in this probe, we actually use a cyber green. Now, I told you at the beginning that cyber green cannot differentiate because cyber green binds to each product and it cannot tell me what is the sequence. So what you do here is that Remember that whenever you do cycling, at the end of the cycle, your fluorescence, which is from the cyber green, will be very, very high. It comes in the y-axis, which is the fluorescence, very, very high. What you do at the end of the PCR, after 30 cycle or 40 cycle, remember that this PCR is done 40 times, 40 cycles, you're going to increase the temperature and you're going to melt the PCR product. Remember that DNA, when it is double strand, whenever you increase the temperature, this DNA start to denature. Whenever it denature, the cyber green will leak out and the fluorescence will go down. So that's why it's called melting, because you're going to melt the DNA, the double strand becomes single strand, and the fluorescence, which was very high, if this is my fluorescence, which is very high cyber green at the beginning, whenever you increase the temperature, the cyber green start to leak from the double strand and the fluorescence will go down again, and it gives you, I call a melting curve. It's called melt curve analysis. Now, how does this help me to understand the genotype? So that's the question I want to ask. So this is, again, I'm trying to explain the same thing. So during the PCR, this is heating, annealing of the primer. And when the synthesis happens, cyber green will bind to the double strand DNA molecules and the fluorescence will increase. And this is what's happened 40 after 45 cycles. This is the end of the PCR from here. So this is a PCR. Denaturation, annihilation, extension, and final extension. And this is your PCR protocol. Now, what you do in high resolution melting, you actually ask what you can do, what is called a melt curve analysis. All what you do, you're going to heat the DNA. So all the cyber green will drop down the fluorescence because the DNA will be single strand. You can see 95, 15 seconds. And then you're going to anneal the DNA. So all the DNA PCR product will be annealed. So cyber green will still go up. So here in this melt curve, you're going to do what is called a melting curve, which is a melt curve here. You're going to increase the temperature from 60. So if this is the temperature, so the x-axis is not cycle number, it will be the temperature, and the y-axis is the fluorescence. So from 60, all the double-strand DNA are actually formed and they are highly fluorescent. And then you go slowly increase the temperature to 95. So you start with 60 and you end up with the 95 degree. What's happened to this DNA? It start to denature, and whenever denature, the cyber green will leak out and the fluorescence will go down. And then you put the camera here. Sorry, let me remove this erase pen. Okay, you put the detection here at the auto exposure when the temperature goes from 60 to 95. And that's why this double strand DNA with so many cyber green will become single strand, and the cyber green will go down, and you're going to get what's called the melting temperature. Midpoint of this is called the melting temperature, or TM. This is the melting temperature. Actually, we use as the midpoint between the graph. Actually, this can help me to understand the mutation. And this is actually, we use the cyber green, which is a modified version of cyber green, which is sensitive to temperature. Actually, the behavior of the line, how it melts, is actually different between different mutation. Remember, whenever you have an A allele, it will bind to a T. If you have a G, it will bind to a C. C binds with the G with 3 hydrogen bond, but this will bind with 2 hydrogen bond. You need more temperature to separate this. So that's why one of the way to understand if you have a mutation A to G is that the G will need more temperature and the A allele will less this temperature. So I can tell from the melting curve that this is actually an AA and this is a GG, and the one in the middle will be the heterozygous AG. And actually you can do the derivatives. You can. This is some of the derivatives يسموها بالعربي الاشتقاق. If you guys remember your math in the past, it can change this graph to look like this. So what you do here, you take a 96 well plate. All what you need to do, just put 2 primers. And there is actually some rules on high resolution primers. It's not like any primers. And then each well, it will give you a graph. All what you need to do, you need to run 3 positive controls. So the only thing you need in melt curve, in high resolution melting, you need to have one sample which you know that this is the wild type, another sample which is heterozygous, and another sample which is homozygous of the mutant version. So the software will tell me that, okay, this is actually the wild type, homozygous, this is the mutant, homozygous, and the one with the double peak is the heterozygous. So any sample unknown, other wells, if you have other wells here, it will see the melted cave. If it looks like this, it will tell you that this is wild type. So from this machine, actually, you can actually genotype, and you need to optimize this one, and you need to design good primers to actually genotype. We actually designed, we use HRM in our labs, and because this is very cheap, and if you have so many samples, and if you want to screen a big population, we, instead of using TACMAN probes, we use high resolution melting and can give you a very good answer. So we had actually one of our master students, she actually genotyped around 2,000 people on a specific mutation that caused some of the liver disease. So we actually have that, and it works very good. Now, the other thing you do in the melt cave analysis is that this is the melting cave analysis, and this is the derivatives. It can tell you whether your PCR gives you a very specific product. In PCR, if you run on a gel, sometimes you get what is called a primer dimers. These primer dimers are the small PCR products that actually, if you use cyber green will bind to this and to bind to this. So it can give you fluorescence from this product and from this product. So in real-time PCR, we always want you to have a very specific PCR product. We don't want to have primer dimers. So whenever you run melt cave analysis, it can tell you whether your primers are good. It gives you specific products, so you're gonna get this kind of graph. So whenever you get double peak, like this is your specific PCR product, and you get a peak at low melting temperature, and these are your primer dimer. So this means you have a very bad primer design, or the concentration of primers are not good. So you need to rerun your PCR using different setup and different primers. So that's what basically the melt cave analysis also can be used for. Now, the other application of this, suppose that you have one of the PCR products that your genotype is increasing in the size. Like this is 100 base pair, your PCR product. And because of certain mutation or insertion in the bacteria, it becomes 150 base pair. So there is an increase of 50 base pair. You can use the melt cave analysis can tell you in the detection, because this will requires more temperature to be separated than this one. So in this protocol, if you have, you're gonna get two peaks. This is for 150 because you need more annealing temperature, and this is for 100. So you can genotype with this. If you only get only hundreds, you can say, okay, this is homozygous 100 and 100. If it is heterozygous, you can say this is 150 or 150 over 100. So again, you can still use cybergreen to genotype. You can genotype SMPs or a size difference in the PCR products. So that's why the second workshop, sorry, this is, okay, so this was the workshop number 1, as I told you, that we're gonna describe about how to design primers, multiplex, and also how you specifically design primers for HRM. There are certain rules for HRMs. So this will be the topic of workshop number 1. Now, the other important topic here that I want to discuss, I'm actually trying to finish fast now, is actually how we're gonna use RT-PCR for quantification. And I can tell you, and I think most of the attendees, they would like to know this more than genotyping and detection, because we use RT-PCR mainly to quantify. And there are different applications. For example, if you want to use DNA, I want to see whether you have virus or not virus, but I also want to answer how much of the virus you have. So viral load, also bacterial load. The other thing you have to see, like if you're doing a transplant, when a bone marrow transplant, or you're doing so much is the mixture, like you're gonna have the recipient cells, individual who has been donated, so you can see how much of this cell to this cell. So I want to quantify. Also, sometimes you get cancer cell. I want to see how much of the cancer tissue you have, like in the blood. So it can tell you what is called the cancer cell load in the sample. So this is quantification. I want to know how much. You can also use messenger RNA expression, microRNA, scripts like this. If you want, I can actually have a very separate talk about microRNA. Or you have what is called long coding RNA. But again, you need to convert them to a DNA with the reverse transcriptase. The other application for quantification is actually if you want to study what is called copy number variant. You want to quantify how much repeats. Sometimes you have a mutation where a part of the genome gets duplicated. So we call them repeats. And these repeats are very important. You can actually use real-time PCR to study repeats. I'm going to talk about it in the coming few slides. And again, in all the techniques, you can use cyber green. So you only need to design primers, or you can use a probe. So you can design primer with extra probe with the fluorescence. You can multiplex this. So both you can use for quantification. Now, what is the principle of quantification and what kind of analysis we do here? As I remember, if I go back in the PCR, as I told you in PCR, if you have a very good efficiency, usually we assume it's 2 because after one cycle, you're going to get 2 copies. So we put 2 to the power of n. And if you start with 1 molecule of DNA, you're going to get 2 copies after one cycle. But if I start with 2 copies of DNA, after one cycle, I'm going to get 4 copies. So the more starting material you have, the more products you get per cycle. So it means, when I say product, I mean more fluorescence. Remember, in cyber green or in the probe technique, after each cycle, you're going to get more fluorescence. So actually, we're going to use this to help us to quantify how much was your starting material. And this will answer all this question: how much messenger RNA you had, how much virus you had, how much cancer cell you had at the beginning when you load equal amount of the sample in your PCR. So that's why if you have this sample, like I have a sample, like it amplified. So basically, if I go back here, so this means if you have a sample which is more concentrated, and remember that whenever I do real-time PCR, I told you that here the camera cannot detect the signal. It needs certain amount of fluorescence to be detected. Now, if I start with more sample, it means that the sample which is more concentrated, it will be amplified much earlier. So if I go here, I put the red color, this sample which is more concentrated, the machine can detect fluorescence earlier because you get more fluorescence per cycle when you have more concentrated samples. So if I go here in this graph, if I put the line, this sample amplified earlier than this sample. So what you do here, you draw a line on the y-axis. We call it the threshold line or the threshold level. So you choose, so if you have like 96 samples, so if you have, this is all my sample, 96 sample, we have so many lines, all of them got amplified. Each one have different concentration. I just put one line on all the samples, and we call this line the threshold line. The moment it crosses, we call this cycle the threshold cycle. Somebody asked me, what is the CT value? CT value is the threshold cycle. And why is that? Whenever you run all your sample in one plate, you need to choose one line. And this will tell me that this sample 20, and this sample is around 30, 29. It will tell me that this sample is more concentrated. So the lower is the CT value, it means the sample amplified earlier because you have more starting material. So if I'm actually studying 2 samples, if I have 2 tubes here, and I have so many messenger RNA, of different genes that are expressed. So if I design a primer that only defines this RNA, and here I have 2 copies of this RNA, this sample is more concentrated than this one for this specific gene, specific messenger RNA. So that's why this sample will amplify earlier. So I can use the CT value to tell me that this sample has more than the red sample. And this is the basic of quantification. And this is actually an example. I'm showing you some of the examples we have. So this is a normalized fluorescence. I'm not going to talk about normalization. Okay, actually, I'm going to talk, but it's a very important topic when you talk about DNA and about analysis of real-time PCR. Now, these samples, all of them, you can tell you that the CT value, if this is the threshold line, the CT of this is around 11. This is around, like, for example, 14. The software will tell you the CT value. It will draw lines for each of these samples. And usually we run sample in duplicates, so which is a good practice for you. So when you have good pipetting, it will give you the same CT value for the average of the sample. Now, one of the problem of this is actually whenever you have a sample, the same sample, I have 2 tubes, and I use 20 microliter reaction in all the samples. And then I start to load my sample here. Suppose I load my sample, which is RNA or DNA, 2 microliter. 2 microliter. But you have some problem with pipetting. Instead of loading 2 microliter, you loaded like 2.1 microliter. So what it means that because the PCR is a very sensitive method, this, although it is the same sample, I'm supposed to get the same CT value, but because I loaded more, the CT value of this sample will show more than this. So what we do here, we actually control for loading. We always run 2 genes, and this is what is called normalization. So you run the gene of interest. If you're running gene expression, you normalize to a gene we call the housekeeping gene. That is gene that is not being changed during the experiment. And the quantity, if you have more input, the gene will be more in that sample. So you always do what is called normalization. And in the workshop, we're going to discuss about different methods to normalize. For gene expression is different, for copy number variant is different, and for microRNA is also a totally different normalization technique. So let's discuss about analysis. And this is actually, I need all of you to focus on this, especially those who are expert in this field. I need you to focus on this because there are different ways to analyze the real-time PCR data for gene expression and for quantification. Now, suppose that I have a study where I'm going to have 2 groups. So I have a group number 1, like untreated and treated group. I want to get a bar diagram with the standard error, and this is my sample size. So this is group number 1, which is the control group, and group number 2, which is the treated group. How many samples I have? I have 1, 2, 3, 4, and running them in duplicates. So this is the duplicates. So usually it's good to run duplicates. So I have 4 samples. So N is 4, and the other group is also 4 samples. What I do, I run for the gene of interest, like this is one of the genes I'm interested in, it's like an inflammatory gene, and then I'm going to run a housekeeping gene in the same plate, 96 well. So if you have space, you can run the gene of interest and you can run the housekeeping gene on all the samples, all the groups, then you can use what is called the delta-delta-CT method. Or comparative delta-delta-CT method. So if you have all the sample, all the groups can be run on the same, in the same plate, not wells, you can assume that the efficiency, why is that? Because the efficiency can be variable between the different plates. So if you run them all in the same plate, the same camera temperature, the same fluorescence intensity, you can assume that the efficiency is the same, and you can say the efficiency is 2. So that's why you can run what is called the delta-delta-CT method. Now, in this example, I still have space. So if I look at this plate, I still can run more genes here. I can run another gene number 2. I can run another gene number 3, gene number 4, and gene number 5. If you have like your sample like 8 sample you have here, because you have 4 for group 1, 4 for group 2, you need to run the housekeeping gene once in one plate. So you can still run delta-delta-CT method. Now, and this is one of the examples, like we measure the inflammatory nitric oxide synthase, and the data you're going to get in delta-delta-CT is a fold expression. And we're going to discuss this in the analysis in the workshop. Now, let's take this example. Now, here I have more samples. Like group number 1, I have 4 times 3, so I have 12 samples. So this is another experiment where my sample is more. Again, I can run gene number 1, all the samples, and the housekeeping gene in the same plate. So if all sample and groups can be run on the same plate, you can assume efficiency again the same, and you can still use delta-delta-CT method. Now, what is the problem here? If I have more than one gene, that means I have to run all the time gene number 2 with housekeeping again, because whenever I do normalization, the efficiency of PCR for housekeeping should be the same for the gene. So you have to run, so it will become more costly for you. So that means that you have to run the same sample for the gene and the housekeeping every time in every plate. But again, you can use delta-delta-CT, but here we can use another method that I'm going to talk about. It's called relative standard method if you want to make it cheaper for yourself. So I'm going to discuss later on about the relative standard method. Now, in this example, I have so many samples. So group number 1, I have like around 48 samples. Group number 2, I have 48 samples. 24 samples, because I'm running in duplicates. So let's say 24 samples. So if you have like so many samples, you have a study where your sample is too many. In this case, I can only run one gene. Whenever I say one gene, I mean one primer, okay? because remember, one master mix means one primer. The problem with this, you cannot run the housekeeping gene in the same plate. So the only thing you can do, you can use TACMAN probes where you can multiplex. So remember, one gene, it gives you green, and therefore the housekeeping gene, you can do another gene. But if you don't have multiplex and you want to use cyber green, then there is another way to do it. Let me just erase the pen. So in this case, if you cannot run all the gene in the same plate, but you can run all the samples and the group in the plate, then you use another method called efficiency corrected delta delta CT, or another name is called FAFL method. In here, you use the efficiency. Instead of using 2 to the power n, you need to get an efficiency from the data of the PCR plates. So I'm going to discuss that in the workshop, how you're going to get the efficiency. And usually we get it from the slope of the diluted sample, serially diluted sample. So again, here is another method called efficiency corrected delta delta CT method. Now, another method you can also run here is, for example, here, in this case, I can run all the groups in this plate. But suppose I actually got this issue with one of our master student. She had the samples like of many, and she cannot run them all in the same plate. So this is group number 1. It means she needs to run another group number 2 in another separate plate. So she has so many samples. In this case, you cannot use delta delta CT. It's become very difficult, and you cannot also use efficiency corrected delta delta CT. For this special case, which is very rare, you can get somebody who's doing so many samples for a rate. You use what is called another efficiency corrected method, but here you can get it from the amplification curve. Remember the amplification curve? This slope, it can tell you the efficiency of the PCR. So you need to run what is called land regression PCR method, and this is actually a software which is freely available. You can do it in Excel, but you can also get a software that can do that for you. So this is again a totally different analysis method that you run for your PCR. Now the other common PCR also method, actually I like to use this more than delta delta CT because it saves a lot of time and money for me. In this case, suppose that I cannot run the housekeeping gene and the gene of interest in the same plate, but I can run all my sample and group in the same plate. What you do here, you run another sample we call the standards. And these are standard of serially you do serial dilution of a sample, and then you're going to get the standard curve. It's like in ELISA, you do what is called a standard curve. You get a linear graph, and then you do the unknown. So from the curve, I can get the quantity from this sample. Now, how to do this? And this is, for example, one of them is actually get the relative gene expression of your interest, and you get the quantity of your housekeeping gene, and you divide them, and you can get actually a good statistics when you do different genes with the relative standard method. Now, how to do that? As I said, you're going to run what is called the standards. So these are different wells in your plate, and you take a sample which is highest concentration. This is the cancer concentration 1, and then you do 1 in 4 dilution or 1 in 5. So you do serial dilution. All what you need to do, you just prepare samples. If this is 1, you do 1 in 4. So you take 1 microliter of this sample plus 4 micro of water, and then it will be serially diluted 1 in 4. And then you know that this is 1 in 4, you can know the relative concentration, which is the relative dilution. If this sample is 1, this will be 0.25 because this is the quarter of that. The next sample will be also 1 of 4 of this 1 of 4. So it will be again 1 over 16. So and so on. So this is like serial diluted sample. So you run these samples, and then you're going to get what is called a standard curve. Okay. So this is the standard curve. You're going to look at the R-squared value, and this is one of the things you check, that it is a linear curve. So you get what is called the CT value. You're going to put a threshold line. This is the CT, and this is the quantity that you're going to get actually from the. So you're going to each of this quantity, you're going to get it here on the y-axis, and then you're going to run them, and you get the standard curve. Now, if you have an unknown samples, so these are your standard samples. Any unknown sample, suppose that this is sample in one of the well, it gave me a value which is here. So the software will get the CT value. From the CT value, if this is like the CT value was 22, it can tell me, okay, the quantity of this sample is this. So each sample, you're going to get what is called a relative concentration and then you're going to give you the value. So what you do, you run a gene of your interest, and this is the quantity you get from the standard curve. And then you also run the housekeeping gene with its own standard curve. And then to normalize, you just divide 1 by 2 and you get the value. And then you're going to get what's called normalized value. And this is what you're going to get in the curve at the end of the day. So in the workshop, I'm going to discuss all of this. So we're going to follow up with this workshop. We're going to do how to set it up. So I'm going to show you how you're going to do delta CT method. And then I'm going to show you how you're going to use FAFL method, which is efficiency corrected delta CT. Remember that you cannot use, this is a mistake as many people, also even scientists. I do review a lot of manuscript I do get from the internet, like many journals. I sometimes see people like making a big mistake. So remember that whenever you do your projects or whenever you do have any experiment, you have to know which method, the correct method to use. Delta CT is the commonly used if you have very few samples. But if you have many samples, then you have to shift with the line reg PCR FAFL method. They are different delta CT method, but it all depends on how many samples, how many groups, and whether you can fill the whole plate or not. And then the other method also I'm going to discuss is the relative standard method. The other way also we want to discuss in the workshop is the normalization. We're going to discuss normalization separately and also from microRNA, which is called the global mean normalization. And this is what we need to discuss, because for microRNA, there is no housekeeping gene. So you'll face a problem to normalize which gene I need to choose for microRNA. So this is something that we need to discuss in the workshop. Now, actually, I have just 2 more slides, and then we're going to be over, inshallah, and we can ask questions later on. Now, this one, I'm going to discuss about how we're going to use real-time PCR to measure what is called copy number variant. Now, let's take this kind of repeat. This is like a DNA chromosome number 2, a region chromosome 2, and this is the mother and the father. And then you have what is called a part of this DNA is being repeated. Same sequence being copied. 1, 2, 3. So this individual, you can say he has 3/3, the genotype. So you have 3 repeats in the mother chromosome, 3 repeats in the father. This individual will have 3, 1, 2, 3, 4, 5, 3, 5. So he's heterozygous. So you have 5 repeats from the father and 3 repeats from the mother. So this is actually what you use. Even people use this technique to do DNA fingerprinting. Actually, if you go to the police now, they do what is called DNA fingerprinting, and they use what is called microsatellite repeat. And those repeats, you can actually use a sequencing machine, Sanger sequencing machine, fragment analysis. But sometimes you do get a problem. If this repeat size is small, you can use HRM, no problem for you. Sometimes if it's a repeat is like a microsatellite, then you can use the Sanger PCR method, which is the Sanger sequencing machine. But if you sometimes you do feel that this repeat is actually very long, it's not a small repeat, it's actually a very big chunk of DNA that's being repeated. And here you're going to face a problem that your fragment analysis, or what is called the HRM, and also there is another technique called MLPA. This is actually a technique we sometimes use for some of the repeats. It does not work in that method. So only way to do it is actually use what is called real-time PCR. And all how you set it up, you only need just to design a primer that binds to the repeat. So the same primer, left and reverse, binds here and there. You can design a probe, fluorescence probe, you can do that. But again, in this PCR, if this is one individual, you get one nucleus DNA. You're going to get a copy from mother and father. You're going to get how many repeats here total? 1, 2, 3, 6 repeats. And this will give you an answer. Now, if I want to do the same for another individual, we have how many repeats here? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And you're going to design the same primer left and right for all of this. All would it tell you that because this has 10, even if you start the same amount, if I use 10 nanogram DNA in all of this and 10 nanogram DNA here, I use the same starting material, but because this has more repeats, it's like you're going to get more quantity than this one. Because you have 10, the primer will bind to 10, here primer will bind to 6 region. So that's why the CT value here will be more than this one. It's like using the quantitative PCR, but here instead of quantifying the messenger RNA, I'm quantifying number of repeats. So again, the same principle, you're going to get the CT value. So if you have less CT value, it means you have more repeats because you're going to get, there's more PCR like starting material because of you have more repeats. But again, this is totally a different method, and you need different normalization, and it requires certain calibration that if you are really interested, I'm going to talk to you. I'm actually currently working in one of the projects using this lipoprotein A repeats. And this is like one of the papers. If you're interested, you can discuss about copy number variant using a quantitative PCR. And it's actually one of the methods that's been used now for clinics to look for the lipoprotein A polymorphism, like copy number variants. And the last thing I want to finish with this is actually some people who are interested in epigenetics, like looking at methylated DNA. Again, in methylation DNA, you need to do before the start of the real-time PCR, you need to what is called bisulfite treatment. Any cytosine which is methylated will be protected. It will not be changed. A cytosine that is not methylated, it will become a T at the end of the day. So basically, this is a chemical reaction you need to do for your reaction that the C will become a T, and the methylated cytosine will not be converted to T. And then you can use a real-time PCR where you can actually, because this is like a C and a T, you can design primers that binds to the C and primer that binds to the T. And then you can actually quantify that. Or you can design a probe with different fluorescence. This will be VIC and this will be FAM. It's again the same principle, it's just how you design the primers or the probe that binds to different genotypes. And you can also quantify the methylation. You can use the quantitative method, and you can also use the HRM for methylation. Because remember, this is a C and this is a T, would have different melting behavior. Same PCR product size, but different melting behavior. It can tell you whether the sample is methylated or not methylated, and you can use HRM. But again, HRM is not commonly used for methylation, but you can still use it. Many people use a methyl-specific PCR for this purpose. So that's the end of my talk. Remember that this was an introductory webinar. Just try to introduce what is a real-time PCR. I'm going to follow it up with 2 workshops on primer designs, and the other workshop, which is more specialized on how you actually analyze the data for quantification. If you have any question, you can always email me. I'm available in the university, and if you have anything that you're interested to ask, you can ask me now, and the floor is open now for everybody.
شكرا لك دكتور على جهدك ووقتك وهذا الطرح الطيب والإثراء الجميل. استفدنا صراحة بهذه المعلومات القيمة، وإن شاء الله للقاءات أكثر في قادم الأيام وفي قادم الورش. مثل ما ذكر الدكتور، حين أي حد عنده استفسار ممكن يسأل على الشات، الدكتور بيقراكم إن شاء الله. شكرا للجميع.
إن شاء الله. So I will go now. I got a question. Just continue. Abdullah Al-Madhani. He asked me, "Can you? Thanks. How about? Okay. Can we have the PowerPoint? Yes, of course. I can send to Yusuf, and he can share to you because I don't have the email of everybody. So then you can say, are you using the same RNA extraction method for both human and virus to ensure the RNA quality? Now, Ibtisam asked me questions on not on real-time PCR, on another method, which is the RNA extractions. Remember that for RNA extraction, it's the method you can use trizol. I mean, it depends on the size of your RNA. Remember, the virus RNA can go to around 30,000 base pair, 30 kbs. You can use trizol, no problem. You can use the RNA extraction kit. Remember, there are 2 methods to extract the RNA. It's using basically the trizol, which is actually helps you to maintain the RNA, and you can actually purify that with the kit. And people nowadays use magnetic like beads to extract the RNA. Again, you can use that for viruses. Now, the problem happens when you have an RNA which is very small, like the microRNA. Then the microRNA, actually there is a modification of that protocol, but you can still use the trizol reagent, but you need to modify it. So it's fine. The answer, yes, you can use that method. I did not get the idea behind the housekeeping gene and why we are running the same amount of wells of housekeeping compared to our unknown samples. Do you need to lead to housekeeping so that we could compare it to the unknown sample? Yes, actually for each sample, because remember that whenever I want to run like sample number 1, I want to measure like gene, like let's say inos. I need to know the same sample, how much was the housekeeping gene. You need to normalize that. And this is what I'm going to discuss, inshallah, in the workshop. Remember that I said we're going to discuss about normalization in the workshop. I'm going to explain to that, the purpose of normalization. So you need, for each sample, you need to run the housekeeping gene. Now, I have another question. Is it quantification until statistic, temperature? Yeah. Okay, what is the time? Okay, then somebody, Mariam Al-Jabri, she asked me, what is the time of your workshop? Yes, again, this will be, again, I'm going to discuss with the people in this platform. Hopefully, inshallah, in September, I'm going to have one workshop. I'm going to update you. Inshallah, we're going to have it in Twitter. So please follow us in the Twitter and we're going to talk about it, inshallah.
Doctor, please.
Yeah.
About ask for Mariam, just follow the Data Academy website in the social media. You can see all information about the workshop. Thank you, Doctor.
So Ruqayya, she asked to share the PowerPoint. Again, we're going to do that. Somebody sent an email. Email. I'm new to the feed. Do we need to use SPSS to do a static? Okay. Somebody actually asked me, do you need to use SPSS? Actually, SPSS is actually a statistical tool. Real-time PCR is actually a method that gives you numbers, data. How are you going to analyze those data? You can actually use SPSS, and if you don't have SPSS, actually Excel is more than enough. Actually, Excel can do all your analysis because most of the time in the real-time PCR, we usually use student t-test or ANOVA. Maybe you can use Mann-Whitney U-test if you have non-normalized data, but usually in real-time PCR, if you're using a very controlled experiment, you can use Excel more than enough. What's happened if you are using a microarray data when you're doing RNA profiling? Here, the Excel might be helpful, but I actually use R for program. SPSS, actually, I don't like it because the graphs that I get from SPSS are not fancy. I like to have fancy graphs. So we can use the R. Actually, I use R most of the time. R can give me very good graphs and very good analysis too. My Twitter account, I think it's there in the data. I can share with you my Twitter. If you just search for Faz Zedjali, sorry, I can't. I'm not a guy on the social media. I'm not into social media that much. So I usually don't tweet much and I don't use too much social media. But you can search around, you can find me. Okay. So we have more. Please let me know when you announce the coming workshop, inshallah. So I think we are almost there. And so for plant, can we use real-time PCR for fragment analysis? Okay, somebody asked me real-time PCR and fragment analysis. Well, fragment analysis is totally different. Fragment analysis, you use the standard PCR machine, and also it's fluorescence based. But you can, to get the same answer for fragment analysis, I already described that you can actually measure repeats and the size variation. But again, there is a limitation for real-time PCR. So fragment analysis is different from that one, okay? Can you please cover more on next version for epigenetics? Shadan, he's asking about RT-PCR on epigenetics. Of course, maybe we can have a separate workshop on epigenetics, because in epigenetics, as I told you, you can use method-specific PCR primers, or you can use a primer that we specifically bind, or you can use HRM. So both of them, actually the principle is the same, but it's just how you design your primer is different. So yes, we can actually maybe try to discuss it when you talk about the primer design. Sorry, I'm trying to, because I have the chat and I have the question and answer. I'm looking in both directions. Why it is difficult to detect parasite with RT-PCR? It's actually not difficult. The only difficulty you do is actually how you do your extraction. I mean, if you have a good extraction method now, and this is something I need to discuss with people, like with you. I mean, if you have, like, for example, a sample that the parasite is very low, and I know people who, like, for example, studying a parasite, for example, in a plant, and the parasite is only one copy. When you do extraction, you make sure the parasite DNA material or RNA material is also included in that sample. And there are a lot of ways to increase the yield of your extraction method. And you need to be familiar with the extraction of DNA. Remember, sometimes we do add glycogen actually in the extraction. Why we add glycogen? It helps in extraction very low abundant DNA. Sometimes we use a foreign DNA like a fish sperm DNA. Sometimes we add during extraction. Why we do that? I think that will help. So RT-PCR is very good as long as you have a very good sample. So that's my answer for you. Okay, now we have, thank you. Can you please cover more in your workshop on how to use epigenetics? Yeah, that's done. I think we are done with the questions.
I think there is no question now.
Yeah, okay. I think I spent a good time. What time is now? Oh, mashallah, almost.
Thank you, Doctor. Thank
Okay.
you for all attendance. Thank you for your time. Thank you. We'll see you in the next workshop. Thank you, Doctor. Thank you to everyone.
شكرا جماعة.
See you, inshallah. مع السلامة.